Right. Okay, thank you for the opportunity to tell you a little bit about what we have been doing over the last few years in my group, looking at clonal plants. So you can see in this slide is pretty much the biological question that we are trying to understand. So this is from Seisuke Kimura, and it's basically a cross-section of a leaf. And what you can see here is like little plantlets that regenerate from the margins of this leaf. Oh, okay. Move forward. Yeah. Okay. So I would imagine the audience is primarily plant biologists, and I guess you are aware that plants differ to animals in several processes, but two of the most important ones from our point of view is sessility, which is the incapacity to escape from stressful environmental fluctuations, and perhaps linked to this is totipotency, which is the capability of adapting a developmental program to reproduce asexually. Okay, and what my group has been trying to investigate is both of these questions and what are the consequences of these biological properties in plants. So most plants reproduce asexually from terminally differentiated somatic cells, and I took this beautiful graphical representation from Keiko Sugimoto's review a few years ago. And what you can see here is that plants can reproduce from, well, asexually reproduce from sexual organs, like for instance from gametes, and this can be done naturally. You can see here some samples of embryogenesis from pollen, but it can be induced as well. And this is the platform that is currently used to generate haploid plants and double haploids for breeding, to accelerate breeding. But for those of you that, like me, may be interested in gardening, you know that you can clone plants just basically by inducing shoot formation from cuttings from the stems or from leaves, which is represented here. And then is another route to generate clonal plants asexually, and is direct embryogenesis. And this could be done in many different ways. This can be induced with hormones. It can be induced from explants, or it can be induced from individual cells, and it can lead to the formation of somatic embryos, and then ultimately plants. So those programs are usually activated through wounding, and I'm not going to get into detail into that particular pathway, but I'm more interested in the developmentally regulated asexual programs, and these can be hijacking or utilizing zygotic transcription factors. And there are very good examples in several plant species. I'm going to give you two examples. One of them is the classic example in Kalanchoe in the crassulaceans group. This is work done primarily by Nila Sinha in her group a few years back. And what she found is that some of these Kalanchoes, that they have this ectopic formation of somatic embryos, they actually are linked to the truncation of a zygotic factor known as LEG1, which is very important for embryogenesis. And some other species, they have a conditional somatic embryogenesis or asexual programs, which are always associated with the truncation of this transcription factor. And we know as well, and as you will see later, that the ectopic expression of these transcription factors in Arabidopsis and other plant species can induce organogenesis, the formation of somatic embryos, and this can facilitate the cloning of plants. Another interesting example is that I highlight here is in oranges. And this is interesting because genetic variation has created insertions of transposons in some genes that encode for zygotic factors. And the one that I'm giving as an example here is this one called RKD1. These RKD genes belong to a group of the RWP-RK domain transcription factors that is exclusively expressed in egg cells and in zygotes. And in this particular example in citrus, the insertion of a mite retrotransposon induce the expression of this RKD gene, and then leads to the formation of polyembryony. So this, you can imagine like an orange with seeds inside an orange and inside an orange, so polyembryony. And this is work that is being done by Mitsuo Omura from Tokyo University a couple of years ago. So these RKD or RWP-RK transcription factors or RKD genes are really interesting because the ectopic expression of these transcription factors lead to the formation of organogenesis or direct organogenesis in different organs. This is work done by Keiji Nakajima in Nara, and he identified mutants that they are embryo lethal. But what is interesting is that the induction of this transcription factor leads to the rapid proliferation of cells in these organs, and then the release of the induction allows the induction of embryogenic programs, which is what you see here in roots. So these are little somatic embryos that develop on the surface of a root. And this allows the direct cloning or regeneration of plants from any organ. So this is an example from roots, but I could give you an example from leaves or stems. So this is interesting because these strategies may allow us to address some interesting questions. So for instance, from the ecological point of view, most plants that they're invasive, they use at least at the beginning or through the spreading of the populations, clonal reproduction as a strategy to spread very rapidly. So these clonal plants are supposed to be genetically identical, but several studies, and this is the one that I'm going to highlight, it goes back 20 years, done by John Parker in Cambridge. He found, and others found, that some of these populations display genetic variability, and this was unexpected. So most of this work in the late 90s, it was done using AFLPs or RFLPs, and these techniques use restriction digestion of DNA, PCR amplification or southern blot, to be able to detect these polymorphisms. And what is interesting is that these enzymes, some of these enzymes are methylation sensitive. So at the time it was not known, but now you can imagine that maybe there is genetic or epigenetic variation in these natural populations that they reproduce clonally. And this is something that we wanted to look at. However, the conventional idea is that clonal plants, when they propagate, produce individuals from any organ. And what I highlight here in color is that these organs will be able to reset the epigenetic information because genetically they are supposed to be identical. So I represent here. So the top of the plant will be green, which is what you expect for a leaf. On the root, it will be a different color that it will reset every time that a plant develops, right? And the same will occur for roots. So if we clonally propagate a plant from a root, we expect that the root leaf will reprogram the whole genetic and epigenetic program in the same way as you expect for a leaf and as well for a root, okay? But the work from John Parker and others suggest that maybe this is not the case. Maybe there is a cellular memory that it can be copied from the explant that we use for propagation or is used for propagation into the progeny. And if that is the case, then the individuals that it will come from this induced organogenesis will be able to maintain some kind of cellular memory into the progeny. And possibly the cellular memory could be propagated asexually. This will be simple to explain, but maybe it could be propagated genetically as well through sexual programs. And we wanted to test this. Before getting to this point, one of the experiments that we did, it was to induce hormones, induce organogenesis with hormones, and this can be done in vitro. What we found is that if we clone plants either from roots, which we call leaf origin or allo, or root origin that we call arrow, and we take the leaves from these plants that we clone and allow to reproduce through seeds over at least 2 generations. Then what we found is that there were stochastic changes in DNA methylation, which is what is highlighted here. Red is high, blue is low methylation, and this is comparing to a standard leaf, right? And if we compare these profiles that we obtain from the leaves of the clonal plants to the normal roots, then there are lots of similarities in the pattern of methylation between these plants regenerated from different organs to roots. And these data suggested that possibly when we use hormones, we end up with progenies or clonal progenies that they have the characteristics of a root. And this is quite unusual, but probably is not exclusive to Arabidopsis, okay? Because Stroud, several years ago, conducted a DNA methylation analysis in rice that it was used for transformation. So they went through a regeneration process. And what he found is that in the plants that they were genetically modified, most of these plants accumulated stochastic changes in DNA methylation, primarily hypomethylation, okay? So which resembles our results. What's interesting is that Anjar in my group took some of these plants, grew them over several generations sexually, and what he found is that either plants that they came from roots or from leaves and regenerated with hormones, they accumulated a lot of pleiotropic developmental phenotypes, what you can see here, okay? And these phenotypes were stable across generations, suggesting that they could be associated with either genetic or epigenetic mutations in these plants, okay? And this is a characteristic that has been observed in other plant species, and I'm just going to give you some classic examples of epimutations that they are induced by hormones or hormone-induced organogenesis. So this is a CNR locus in tomato, this is the karma locus in oil palm, and so on. And all these examples are in general associated with hypomethylation of sequences, usually transposable element sequences, okay? So then at this point we thought, well, maybe there are 2 possibilities here. One is that the hormones can induce this hypomethylation, irrespectively of the explant that we use, or that the hormones induce a developmental program which causes this epigenetic change, which can be propagated later on. And this is interesting because Elliot Meyerowitz and Sugimoto, almost 12 years ago, realized that when we take Arabidopsis plants, we induce callus formation, this hormone-induced organogenesis that comes from the callus has the typical signatures of a root differentiation program. And this was a beautiful experiment that they designed using different markers, GFP markers. And these markers are specifically expressed in roots. But independently of the explant that they were using, when hormones were applied to induce these somatic structures, these callus, then this led to this ectopic activation of these markers in these tissues. So it may be possible that hormones induce a developmental program, in this case roots, and then roots change the epigenetic makeup of these cells, and this change can be propagated clonally. To address this question, Anjar in my group developed a strategy, and the strategy is very simple. So we take plants, and then we clonally propagate them from either leaves or roots using a hormone-free system. And then once these plants were clonally propagated, we will sexually propagate them at least over 2 or 3 generations to avoid any maternal effects on these clonal individuals. So we call these lines leaf origin lines for LO, if they originated from the aerial tissue, or root origin lines if they originated from the root tissue. Okay, and just to give you an example of how this works, so we have transgenic lines. We can induce this RWPRK transcription factor, and this can induce the formation of somatic embryos on the surface of the leaves, which we can rescue with microneedles into normal media, no hormones, and then allow the roots and the shoots to form, and then transfer it into soil and propagate them sexually for 5 generations, and the same for roots. Right? So what was interesting is that when Anjar completed the analysis, the first thing that he looked at it was epigenetic changes. And this represents a little bit of the data that he found. So each one of these columns is an individual line that is being cloned and propagated over 3 generations, 10 plants pulled together. Okay, it's not individual plants. And we are comparing the methylation data, methylome data to leaves, normal leaves. Okay, and what you can see here, this group is 3 root samples. Roots in Arabidopsis compared to leaves are significantly hypomethylated. That is what you see in blue here. There are around 200 to 400 hypomethylated regions, or more, depending on the threshold in the genome. But what Anjar found is that when you clone plants from leaves and you compare the leaf methylome and the root methylome, this pattern, it did look pretty much like a normal leaf and a normal root. So suggesting that cloning from leaves, the programs, the epigenetic level, they are as you would expect. But when you clone from roots, those plants that they were cloned from roots, both leaves and roots have very similar epigenetic makeup, suggesting that when we clone from leaves, we get the expected program for leaves and roots. But when we clone from roots, we obtain plants that they actually have the unexpected program because they have these root epigenetic marks in the leaves. Right? And this is heritable because we have been looking at these plants 3 generations later. So it's being able to pass sexually. Which cell in the root contribute to this? Well, we suspected that probably not all the roots in all the cells in the root will contribute to these clonal plants. So for that, Matt Nice in my lab conducted a confocal analysis looking at the cells that respond to the somatic or lead to the formation of the somatic embryos. And he came to the conclusion that the sub-epidermal cells were the primary contributors, what you can see here, to the formation of the somatic embryos and the roots. So to get a genetic handle on this, or genetic handle, then we compare the methylome of plants that were cloned from leaves or from roots with plants that were cloned from individual cell types within the root. And for that, we use different GFP markers for different cell types. So when we compare the methylation data for these populations, right, they were not individual plants, they were populations. We found that all the plants that they were cloned from roots, and these are leaves, all that, they were primarily or resemble significantly to sub-epidermal cells. So this suggests that even when we induce these somatic embryos in our system, most of the cells that contribute to these clonal plants are sub-epidermal. Okay. So we have plants that they have somehow inherited these epigenetic marks. Are these marks stable across generations? So the answer is yes, because we can propagate them just by selfing over multiple generations. But to test really if these are robust, what we have done is generate reciprocal crosses between Colombia that has not been regenerated with plants that they came from roots. And what we found is that, in fact, the F1 hybrids, irrespectively of the transmission, maternal or paternal, had an intermediate level of DNA methylation, suggesting that the chromatids that they were inheriting the hybrid were able to maintain the DNA methylation status that inherited from the parents. And even so, if we look at F2 progenies from these individuals, what you can see here better probably, is that if you look at the F2s, what we see is that the individual plants, in this case, each bar is an individual plant, segregated for the methylation status of the different regions of the genome, suggesting that these epigenetic maps are really stable. They can be inherited by selfing, and they can be inherited after crossing as well. So what are the consequences of this? Well, the consequences that you can observe in this image is that there are significant changes in growth. And this represents different environmental conditions, and they're environmentally sensitive. So plants that they are originated from roots, this is the third generation of selfing, they are significantly different from the controls and the plants that came from leaves, and this could be exacerbated just basically growing the plants in long days. So to get a better idea, what are the changes that causing these phenotypes, we conducted a transcriptome analysis. And when we compare the transcriptome of leaves of plants that they were, or lines that they were cloned from leaves or roots, we found that there were around 750 to 1,000 genes that they were affected between the plants originated from leaves or roots. Remember, these plants are genetically identical, epigenetically different. And around 25% of these changes, or the genes affected, they had an organ preferential expression. So what it means is that they were exclusively expressed in roots or in leaves in a normal plant. And when we carry out a Go analysis to get an idea of the function, and to our surprise, most of the genes that they were affected, they were related to response to the environment, in particular to microorganisms and immune responses, as you can see here. So this gives us an idea that probably some of these plants that we clonally propagate from different organs, in particularly for roots, may be affected in a particular biological function, in this case, probably relationship with microorganisms. But to get a better idea, then we conducted a network analysis. And out of that analysis, what we found is that there was a specific cluster that it was significantly affected. And this cluster, as you can see here, and in yellow is all the genes that are upregulated in that cluster in the clonal plants. These genes were implicated with a negative regulation of biotic stresses, in particular relationship with microorganisms. So if this is the case, then we would expect some phenotypes when plants are being challenged to pathogens, for instance. So this is what we did. So we took plants. They were clonally propagated from leaves and roots, and then exposed or challenged them with Hyloperonospora infection or Pseudomonas infection. And what you can see here is basically that the plants that the lines that they come originated from roots, they are highly sensitive to Hyloperonospora. This is not the case from the plants that they come from leaves. And when we could look at Pseudomonas, we see something similar. So they are more sensitive to the controls, suggesting that these plants that came from roots, they are immunodepressed and they are not able to respond in the right way to pathogens. So when we carry out this analysis, then we realized that maybe we should be looking as well as beneficial microorganisms. So for that, we conducted a microbiome analysis in the plants that were cloned from lines that were cloned from leaves or roots. And what you can see here in this PCA is basically the differences in the communities, bacterial communities. So you can see that the ones that come from leaf, they're relatively similar to the non-transgenic or the non-clonal plants, the controls. But the plants that came from roots, they were significantly different in terms of the communities compared to the other plants. So suggesting that the epigenetic changes that take place or they are inherited in these clonal plants are able to influence significantly the response of the microbiome in these plants. And then we use standard assays with GFP label beneficial microbes. This is Bacillus ameloleukophaciens. And what you can see here is the colonization in roots, immature roots, and you can see control wild type, standard colonization the plants from leaves, but very poor colonization in the plants that come from roots. Okay? So this suggested that there are phenotypic consequences that are significant on those plants that come from different organs. So to get some idea of the molecular mechanisms, we've been doing some work trying to understand where are the regions that control these phenotypes, and this is work done by Nasim Hussein in my lab. And what we have realized is that most of the regions that they are targeted by epigenetically are intergenic regions, and most of these regions act as distal-regulatory elements. So this is one of the regions that Nasim has characterized. Is upstream of a gene that is usually expressed only in roots or in pollen, and when she hypermethylates this region using inverted repeat hairpins, then she gets this complex plethora of phenotypes caused by the misexpression of the gene. She has done another gene that is 3 kb, another intergenic region that is around 3 kb upstream for another transcription factor known to be implicated in abiotic and biotic responses, and then the hypermethylation of this region caused this plethora of abnormality in the progenies. This is caused by the ectopic activation of this transcription factor. So in summary, this suggests that some of these changes that they are transmitted to the clonal progeny based on the cellular memory are able to are somehow targeting intergenic regions of the genome, and these intergenic regions, or some of these intergenic regions, play an important role in the fine-tuning of some of these genes that they are important for organ-specific responses. So this is interesting, but what does this mean biologically? Because the only thing that we have done is just basically clonally propagate plants. And plants that reproduce asexually in a natural environment, they are not like the plants that we are growing in the laboratory. So we started to wonder if there were, if it was possible that there was an adaptive value to these responses in plants, right? And if these adaptive responses could be heritable. So to be able to test this, we use the same approach, but what we did is to expose the plants that they were about to be clonally propagated to biotic and abiotic stresses. And this is work done by 2 students in my lab, Ham Opasafian and Hadi Lanaputra. So I'm going to summarize all this work, but when we clonally propagate plants, they were exposed to biotic and biotic stress, and then propagate them over 3 generations, and then analyze them molecularly. In this case, I'm going to show you data for transcriptome data. Then we compare the transcriptome data of these plants, not challenged to any stress, okay, to the controls, okay, to address the question, will these plants be prime or will remember the previous stress, okay? And the answer is yes. So when we look at transcriptional changes, which here I'm not showing you the direction, I'm just showing change. Then what we find is that when we expose plants to abiotic stress and then we clone plants from there and propagate them over 3 generations, we look at the transcriptome in leaves, all this work is in leaves, then what we see is that the plants that they were exposed to soil have acquired a lot of transcriptional changes in leaves later on, but not much when they came from leaves. Maybe I said leaves before, but came from roots, okay? But not major changes when the plants came from leaves. And when we use cold, then the roots were not very good in creating some transcriptional responses in the progeny, clonal progeny, but the leaves were really good in causing significant changes in the transcriptome. This is in the order of several hundreds to 1,000 genes affected. And when we look at biotic stress, both leaf origin lines were very good in inducing responses, but the lines that came from roots, they were not very efficient in inducing these responses. So this suggested that these plants, that they are clonally propagated and they are exposed to stress, they can somehow acquire new molecular variation, and this molecular variation is underpinned by the cellular memory origin. Depending on the cell, somatic cell that is used for the cloning, then this will determine the response. So we decided to test this a little bit more from the phenotypic point of view. And then Hadi and Ham, they've been looking at the response of these lines that we have clonally propagated from leaves or from roots after exposure to abiotic stress. And what they have found is that when plants are exposed to cold, the lines that they originate from leaves are very good in survival to freezing, which is one of the phenotypic assays, classic for Arabidopsis, but the ones that come from roots are not very efficient, which goes in line with the transcriptome changes that we have that I just showed you. And when we look at salt tolerance, the plants that come from the lines that come from roots are relatively efficient in having enhanced tolerance to salt or to high salinity, but the ones that came from leaves are not very efficient in that response. So which goes in lines with the transcriptome data that I showed you earlier. So to get an idea of what are the molecular mechanisms in place in these cases, then we conducted a methylome analysis. So we'll skip all the heat maps and all the other epigenetic data, but what I can tell you is that most of the regions that we identify are intergenic regions. So intergenic regions are primarily targeted epigenetically. We have started to characterize some of these regions. I'm going to show you data for one of them. This is a differentially methylated region, which is upstream from a transcription factor known to be important for tolerance to abiotic stress. And what we have done is to direct a hypomethylation of this region using inverted repeats, and this has a major effect on the response, in this case, to salinity. And this gene, well, the gene that is regulating this region is this one, TCF1. TCF1 is tolerant to chilling and freezing 1, which is being characterized by CLE's lab a few years ago. It's strongly induced by cold, and mutants, loss of function mutants in this gene cause an effect on responses to freezing. Okay, so this suggests that some of these intergenic regions are specifically targeted by different biotic and mediated stress to be epigenetically modified, and basically cloning, in this case, using direct-organogenesis, enables the inheritance of some of these marks to the offspring. So just to wrap up, what we have found and others have found is that clonal propagation can generate novel phenotypes, that they are not present in founder cells. Some of the causes could be genetic, but we have evidence that there is a large epigenetic component in the creation of these phenotypes. These novel phenotypes are in fact caused by the incomplete reprogramming of these epigenetic marks in the progeny, which suggests that there is implants, there's a good evidence for cellular memory to be passed on over multiple generations. This could be the case in animals, but it's much more difficult to test. And what is interesting biologically is that there is a potential adaptive value in natural populations, and of course, it's a direct biotechnological application for this. And I just would like to finish thanking everyone in my group that's been contributing to this, which is being highlighted in blue. But the key scientist that been contributing to this is Anjar Wibowo. He was a PhD student in my lab, then he went to Detlef's lab, and now he's back in Indonesia running his own group. Claude Becker was in Detlef's lab, and now he's in GMI in Vienna. This work could not be possible without the input of Detlef and his group. extremely supportive, and several other collaborators helping through some of this work and the support from different organizations. And I just would like to wrap up thanking you as well for being here, and I will be happy to answer any question. And if you have anything that you want to discuss with me, or you're interested in working in my lab, please email me on the address that I show you here. Okay.
Great, thanks very much, Jose. So let's start with a question from Catherine Evans, and this kind of is getting to where you were going. I think this is the extension. So how can these differences in epigenome and phenotype that you see in these clonal plants, how might this influence clonal propagation in horticulture or something like that?
The answer is we don't know, but I think my guess is that it's big. Because when I talk to growers, or I talk to small companies that they use clonal propagation for horticultural material, they have a lot of problems. Instability of phenotypes, loss of resistance in some strains. So I think this is just the tip of the iceberg, but because we did not have the means to address this in some of these particular crops, we don't know. But I think my suspicion is that it is quite big.
So you might propose that you could treat a plant at the start with a stress and then clonally propagate, and you might be able to ensure that that crop, that plant is more resistant to whatever you're interested in over multiple generations.
That would be great, but I would imagine it's not as simple as that.
Okay.
But yeah, I think potentially, having the right inducer and having the right, sort of conditions, it may be possible. I think it's predictable. I think it is possible, possibly, to predict it.
Okay, so we have a question from Lawrence Bindschelder, who's at Royal Holloway, and he asks if you've looked at the difference, whether there's a difference in ploidy levels in plants generated from the roots or the leaf tissues.
Yeah, good question. No, but we have sexually propagated these plants, so we haven't seen evidence of seed defects or infertility, which is quite common when you get changes in ploidy. And we have done a lot of sequencing. So we have data that suggests that ploidy is not a problem. We have done some markers as well, synastry lines, to look at the ploidy to see if there are changes. We have not seen changes. Yeah.
Okay, so a question from Carmen Escudero-Martinez, who's in Dundee, and she asked, what type of microbiome members were differentially abundant in the root-derived clonal plants? And
yeah, good question. I'm not an expert in that, but it could be the Ficumyces. I don't remember that, and some Bacilliae. Sorry, I'm not a microbiologist. Have a look at the paper.
So she asks, is that work published?
Yeah, PNAS a couple of years ago. Sorry, I should have said that.
Okay, that's good to know. Okay, so a question from Anja Barman, and they ask, can clonal variations further revert back to the founder plant, or is it always a unidirectional process?
Good question. I think it could, but I think sexually it will take several generations.
Okay.
We are now in generation number 14, and it's still 70% of the marks are still there, but I think this could be eroded with sexually.
Okay. So another question from Lawrence, actually, so at Royal Holloway. So they ask, in palm tree, I don't know if you know much about palm tree, clonal propagation is a huge issue as it generates mentoled fruits. I don't know what that means.
Yeah,
and that's due to transposition. So do you have an explanation for this from what you know in your system?
Yeah. The mantle phenotype is associated with the hypomethylation of retrotransposon in the first or second exon of the mantle gene, and this changes the splicing. And this is work done by Ken P.O.B. and Rob Martinsen. in science, and this is what similar to what I saw you, that there's hypomethylation when you use hormones.
Okay,